Hey guys, welcome to our unit 3.4, photosynthesis. So this is one of the big topics in AP Bio. Photosynthesis gets a lot into details, especially for the light reaction, not so much for the Calvin cycle, but there is a vast amount of details that we must know for the light reaction and not so much for the Calvin cycle. So as I'm going through this lecture is probably going to be one of the longest lectures. I don't know how long it's going to be just yet because I'm just starting my recording, but I'm going to go very details. For some things, I'm going to go above the scope of AP Bio just for you guys to have like a good understanding of the process. And then as we talk in class, I'm going to tell you some of the details that you do not need to memorize. But I'm going to go a little bit more in depth than College Board requires for me to do. So let's start off with just like a basic definition of photosynthesis. And I trust that this is information you guys already know. So photosynthesis is a process by which light energy is converted into chemical energy. Photosynthesis is a metabolic process that is conducted or performed by autotrophs. So not only plants, so photosynthesis is not exclusive to plant organisms. However, any organism that has a chloroplast or has chlorophyll and is capable of photosynthesizing, this process is also occurs. So we don't say that photosynthesis occurs in plants only, but we tend to say that it occurs in autotrophs. So specifically photoautotrophs, so organisms that can actually capture sun's energy and use that to produce food. All right, so I'm gonna go quickly through this because I know this probably is a waste of your time because you already know what these things are. What is an autotroph? Autotrophs are organisms that make their own food. So the food that they make are organic materials, usually carbohydrates, and they make this food from simple substances in their surrounding. So photosynthesis is a and I'm going to pause right here and you should be able to determine what type of metabolic reaction it is. So if it is using simple materials and it's using simple materials to build more complex products, then you should know that that is a anabolic reaction. And anabolic reactions, they consume energy in order to produce complex products from simple reactants. So I want you guys to remember that from our very first topic in this unit. And then heterotrophs are organisms that are unable to make their own food, and therefore they have no option but to consume other living things in order to gain food or in order to gain energy. The evolution of photosynthesis. So photosynthesis first evolved, or it is believed that photosynthesis first evolved in prokaryotic organisms, specifically in cyanobacteria. So this is the earliest type of prokaryotic organisms to exist, and these organisms were capable of photosynthesizing. They were able to oxygenate the atmosphere in early Earth, and pretty much they are responsible for the atmosphere becoming more habitable to other organisms because they provided oxygen to the atmosphere. Prokaryotic photosynthetic pathways were the foundations for eukaryotic photosynthesis. So, like for example, more complex organisms like plants evolved from bacteria that were once able to photosynthesize. And then you guys should recall the endosymbiont theory. So I want you guys, as you're reading this slide or you're listening to this part of the presentation, I want you guys to be able to connect those two concepts. All right, so let's talk about the site for photosynthesis. So the site or the place or the location of photosynthesis. So where is photosynthesis taking place in these photosynthetic organisms? The primary site for photosynthesis in plants occurs inside of the leaves, inside of special cells inside the leaves. And then organisms that are not plants also do have cells that contain chloroplast and that contains their chlorophyll. And that's where photosynthesis will take place. And I'm going to be referring to plants a lot, and I'm sorry if I do so a lot, guys, but I just want you to remember that not only plants photosynthesize. So even though primarily plants are, they make up the abundance of photosynthetic organism, photosynthesis is not exclusive to actual plants, but bacteria, specifically cyanobacteria, can photosynthesize. So photosynthesis occurs primarily in the leaves. And inside the leaves, there are organelles called chloroplasts, and these are the organelles that are responsible for photosynthesis. Where are chloroplasts found? They're primarily found in the mesophyll cells, and the mesophyll cells contains most of the chlorophyll and chloroplast, and that is primarily where most of the photosynthesis occurs. In addition to that, you have other parts of the leaf that also assist with the process of photosynthesis. So, for example, you have the stomata, and stomatas are pores or openings that are surrounded by two guard cells. And stomata allows for gaseous exchange in the plant. So, gaseous exchange meaning that it allows carbon dioxide to come in for photosynthesis, and a byproduct of photosynthesis is oxygen gas, and oxygen gas is allowed to leave the plants once that gas is produced at the end of the light reaction. Now let's talk about the detailed structures of the chloroplast. So remember, chloroplast is a cell organelle that is found in autotrophs or photosynthetic organisms, and this is the site of photosynthesis. So this is the organelle in which photosynthesis occurs. Now, what are some of the key structures of the chloroplast that assist with the process of photosynthesis? So we have the stroma, not to be confused with the stomata. The stroma is a fluid-filled space. So it is an aqueous, which suggests that there is fluid or there is water-like substances. So there is a space that is filled with water or filled with fluid, and that is called the stroma. So I oftentimes like to describe the stroma to students as this is not like a physical structure. However, it's like a space inside of the cell. Inside of the chloroplast. So think of the stroma of the chloroplast like the cytoplasm of a cell. And the stroma would be all of this space right here that does not have like a physical structure. So it's just like a fluid filled space. So that's our stroma. And then in addition to the stroma, we have other structures called thylakoids. And thylakoids are very vital. So, for example, this is a thylakoid, this is a thylakoid, this is a thylakoid. So these are the stacks are all called thylakoids. So they're stacked like, I don't know, pancakes or like pennies, or their descriptions vary for these structures. So these thylakoids are pretty much where the light reaction will occur. And these thylakoids are usually stacked on top of each other. A stack of thylakoid is called a grana. And then inside these thylakoids, we're going to have chlorophyll. And chlorophyll is the green pigment that is responsible for pretty much trapping sunlight. So this pigment is very important, and this pigment plays a very important role in our photosystems, and we're going to be discussing our photosystems pretty soon. All right, so here's something else that you guys should already know. The formula for photosynthesis. So photosynthesis is a metabolic reaction that occurs in autotrophic organisms. And like all metabolic reactions, there are reactants and there are products. And we can determine what type of metabolic reaction this is, if it's a catabolic or anabolic reaction, by looking at the simplicity of the reactants versus the products, and we can determine what type of metabolic reaction it is. So in this case, we have simple reactants such as carbon dioxide and water. And we have complex products such as high energy molecules, such as glucose, and then we have oxygen. So this is telling us that this is an anabolic reaction because these simple these simple reactants were used to build complex products, which means that photosynthesis is also a huge energy consumer. So a lot of energy is actually invested in this reaction in order for the reaction to occur. So not a spontaneous reaction. Photosynthesis is not a spontaneous reaction, and those words should ring a bell by now. All right, I don't want to spend too much time on this. If you think you need to review this slide, you can go ahead and do that on your own. I trust that you guys already understand this part of it. All right, so photosynthesis, during the process of photosynthesis, water is split. So H2O is split into hydrogen and oxygen. The oxygen is not needed for photosynthesis. The hydrogen is very important. However, this oxygen will combine with another oxygen to form two oxygens, and that will give us oxygen gas, and that oxygen gas leaves as pretty much a waste product for cellular respiration, and sorry, a waste product for photosynthesis. And once that oxygen leaves, and that oxygen enriches our atmosphere, and thus why other life forms can exist here on Earth. All right, so I want to get into a little chemistry. So this is like basic baby chemistry. So we're going to talk about reactions a little bit. So we're going to talk about redox reactions. Read or the red, the R-E-D part of the word is for reduction reactions, and the OX, the ox part, is for oxidation reactions. And we're going to discuss what those two things are. And I hope that you guys cover this in chemistry. If you don't, it's fine. It's a very simple concept. So the process of photosynthesis and also cell rep is pretty much based off redox reactions. And these are reactions. These are two different reactions that pretty much involve the transfer of electrons from pretty much from one molecule to the next. That's what redox reaction is. So if a molecule is gaining electrons, then we say that the molecule is being reduced, or that is a reduction reaction, which is very contradicting. The word is contradicting because you're expecting if something is being reduced, or if it's reduction, then it's losing. But actually, reduction is gaining electrons, so it's becoming more negative. And oxidation reactions are pretty much the opposite. So oxidation reactions are actually losing electrons. So it's becoming more positive. So again, redox reactions, and let me just grab my highlighter right here. So a molecule, or in a reaction, molecules can become reduced, which means that they are gaining electrons, or molecules can become oxidized, which means that they are losing electrons. So the ox means oxidation, and that means that they're losing electrons. And that's going to make sense to you in a bit when we talk about NADPH, whether it's gaining a hydrogen or if it's losing a hydrogen, and what is actually happening to the molecule. Is it becoming reduced, or is it becoming oxidized? All right, so in photosynthesis, the electrons are transferred from hydrogen. So the electrons are transferred from hydrogen when the water splits, um, and they're added to carbon dioxide. And then you can imagine, if carbon dioxide is gaining hydrogen, then that's forming a sugar. So it means that because it's gaining hydrogen that has an electron, then this molecule is becoming reduced. So therefore, photosynthesis is pretty much a redox reaction. And that's just an example, but there's a lot more detail to it. Oh, and here's a nice slide. I forgot I had this slide up. So here's a slide, and this is a nice way for you guys to remember the redox reaction, what it means. I don't know if you guys covered this in chemistry. If you did, you can just skip over this part. So oxidation, remember, oxidation is losing electrons, and reduction is the gaining of electrons. So if a molecule is gaining, if a molecule or a compound is gaining an additional atom, it means that it is being reduced because it is gaining. And if it comes with that atom, it has electrons, so it's gaining electrons. And if for example, if a compound is becoming more simple, it means that it is losing its electrons. So it has lost some atoms, so it's losing electrons. A nice way to remember is oil, rig, or Leo goes grr. So um, oxidation is losing, while reduction, I should have my pointer. So oxidation is losing and reduction is gaining. So that's what it means. Or just a fun way to remember it. So for example, if we were to look at water, and I'm sorry if I'm spending too much time on this, guys, you can skip over this part if you don't need to know this. All right, so let's look at carbon dioxide. So carbon dioxide in photosynthesis, carbon dioxide gains a hydrogen to become a more complex molecule, which is glucose. So by gaining hydrogen, it has been reduced because it has gained additional electrons. So it's more complex. While water, for example, in photosynthesis, water is split into hydrogen and oxygen. So water pretty much at the end, the very end of the reaction, we only have the oxygen and that hydrogen went to form the sugar. So it means that this water has been oxidized. So we have it has lost its electrons. All right, so let's talk about the stages of photosynthesis. So just for you guys to know, we're going to go a lot more in detail with the light reaction than we're going to go with Calvin cycle. And that's because College Board does require you guys to know the light reaction in a lot more detail than you have to know the Calvin cycle. The Calvin cycle is pretty detailed, but College Board over the years, they've kind of just like watered this part down. So you don't need to know the details of the Calvin cycle. However, we do need to know a great deal of info for this light reaction. So there are two stages of photosynthesis. We have the light reaction and we have the Calvin cycle. The light reaction is also called the light dependent reaction. And this part of the reaction has to occur in the presence of light. So there has to be sunlight or there has to be a light source. In most cases, the light reaction occurs for plants specifically. The light reaction is occurring in the daytime, or if the plant has an artificial source of light, then that could occur anytime. But the light reaction does actually require light. The Calvin cycle, which is the second part of the reaction, the Calvin cycle is also named the light independent reaction. And as that name suggests, it is independent of light. So that part of the reaction can occur pretty much any time of day. Regardless of whether or not light is present, it doesn't matter for the Calvin cycle because it does not need light. And here's a cool thing. Because the light reaction requires actual sunlight, that is the photo part of the reaction. And then in the Calvin cycle, guess what? Glucose will be made. So glucose is synthesized during that part of the reaction, and thus the name photosynthesis. All right, so now let's talk a little bit about light, being that light is so crucial for photosynthesis, specifically for the light reaction. Light is an electromagnetic energy that is made up of particles of energy, and these particles are called photons. And these particles, they travel as waves. And light is measured in wavelengths, and a wavelength is the distance from a crest on one wave to the crest on another wave. The entire range is known as an electromagnetic spectrum. Um, and electromagnetic spectrum runs from about 380 nanometers to 750 nanometers of visible light. Short wavelengths equal high energy. So the shorter the wavelength, the more energy. The longer the wavelengths, the less energy. When light interacts with matter, light can either be reflected, transmitted, or absorbed. And speaking of absorption, pigments are able to absorb visible light. So when we talk about like pigment, any pigment at all, um, so for example, for this specific case, when we talk about chlorophyll and all its accessory pigments, chlorophyll is able to absorb light. The color we see is the reflected wavelength of the light. All right, so now that we know that pigments are able to absorb light at different wavelengths, let's talk about photosynthetic pigments. So photosynthetic pigments, the primary ones are chlorophyll, primarily chlorophyll A. Um, so chlorophyll A is the primary pigment, and this is involved in the light reaction, heavily involved in the light reaction. And it is a blue-green pigment. And we also have chlorophyll B, which is an accessory pigment. So this is an accessory to chlorophyll A. And it absorbs light at the yellow to green spectrum. And it is the yellow-green pigment. All right, so this little graph right here is pretty much showing you the absorption coefficient and the different wavelengths of light. And we have our key right here that shows chlorophyll A, chlorophyll B. So let's look at chlorophyll A, for example. So chlorophyll A is absorbing light at around this part of our spectrum, and it is the lowest in the green yellow region, and it is also a little bit high in this reddish region right here. Wherever it is the lowest, that is where it is reflecting light. So it absorbs light at this wavelength, and then wherever it is the lowest, that's where it pretty much reflects light. So when we see a green leaf, or when we see the pigment appears to be green, that is the color it is reflecting, but not the color it is absorbing, if that makes sense. All right, so in addition to those pigments, we also have carotenoids, and carotenoids are additional pigments. So these they broaden the spectrum of colors that drive photosynthesis. So these are yellow, orange pigments, and then also we have photoprotection. So the carotenoids, they do serve a role in what we call photoprotection, and it pretty much absorbs and dissipates excess light energy that could pretty much damage the chlorophyll. So any high energy or too much photons coming in that could damage the chloroplast, and it's responsible for doing for pretty much absorbing that and not allowing for chlorophyll to absorb all of it. All right, here is a very quick FRQ question. I'm sorry guys, I don't want to make this video too long, so I'm just gonna post the questions on here, and then you guys can pause this part of the video. The answer is already there, and you guys can check the answers to see. All right, guys, so here we are. The light reaction. So remember, photosynthesis is divided into two reactions. We have the light reaction and we have the Calvin cycle. The light reaction requires a lot more detail. So College Board does require you to know this in a lot more detail. So we're going to go into detail with this one. So, all right, brace yourselves. Here we go. The light reaction. All right, by the end of this part of the presentation, you should be able to explain this diagram here and what's happening here. All right, so here's an overview of the light reaction. So the light reaction is occurring inside the thylakoid. So here, remember these stacks? One stack called a grana. So what happens is two things will come in to the thylakoid. And what does the thylakoid contain? It has the pigments. These are chlorophyll, and these are the pigments that can trap or absorb light. Light comes in, and water also comes in. When light comes in, a few things will happen, and we're going to get into the details of it, but just an overview. Light comes in, water comes in, or end product will be oxygen leaving, and oxygen is a byproduct or a waste product of photosynthesis. So oxygen leaves, however, the hydrogen stays behind, and that hydrogen will be used to reduce NADP, remember our redox reactions. So that hydrogen is now attached to our NADP. Here it is. And then we're also going to have ATP being produced or synthesized by ATP synthase. And then once we have these two guys, ATP and NADPH, then that's pretty much the end of the light reaction. And these two products go to the Calvin cycle while oxygen leaves. So that's pretty much an overview. And then now we're going to go into the intricate details of how do we get here to these two products. All right, so key thing, this is occurring inside the thylakoid membrane, in the photosystems, and solar energy has been. So in the light reaction, chemical energy exist in two forms. Or in simple terms, there are two chemicals that are going to be produced at the end of the light reaction. And that chemical is going to be a reduced NADP. And by reducing, remember, where it's gaining an electron. And what is that electron? This is hydrogen. So NADP will become NADPH. And then also ATP will be synthesized. Um, and this process is accomplished by using light energy, so that's the sun's energy, the photons, to excite electrons. All right, so let's talk about how or why light is so important to chlorophyll. And I believe that this was one of the questions that you guys were asked on one of the previous FRQs that we covered. So light is pretty much very important to chlorophyll because the light is pretty much used, or the chlorophyll traps the sunlight, and then that sunlight is pretty much used to excite an existing electrons. And what are the sources of these electrons, guys? The source of the electrons are water, and that's what plants need water for. They need water to drive this process. They don't need the oxygen, they just need the hydrogen. And hydrogen contains one electron and a proton, so they just need that electron to pretty much to catalyze or to drive this process. So here are the details. So we have chlorophyll, and remember we said these pigments are able to absorb light energy. So the chlorophyll absorbs a photon of light, and then that photon of light, once it hits that chlorophyll molecule, it is used to excite an electron, and these electrons are coming from water. So it is used to excite an electron. Because electrons are unstable, they fall back to a ground state, so they become grounded again, so they fall back. And as they're doing this, they're losing heat, and then also it's losing that light energy, so that fluorescent is being emitted. So that's pretty much why light is so important to chlorophyll. So it's releasing that heat energy, and also it is releasing photons. All right, so if that made very little sense to you, it's going to start making a lot more sense once we start piecing everything together. So I'm explaining every little thing in chunks, and then we're pretty much going to be painting the bigger picture as we go along. All right, so let's talk about photosystems. Let's look at this picture that I have here. So if you have not figured it out, this is the thylakoid membrane. You see how this looks very similar to a cell membrane in which we have the phospholipid bilayers, and then we have various proteins embedded in the membrane. So as I'm explaining the light reaction, I want you guys to have a clear picture as to where this is occurring. So thylakoid, these are the stacks, um, are the pennies or the pancake looking structures, and along the membrane of the thylakoid is where the light reaction is occurring, or where these reaction centers are. So as you can imagine, with any membrane, you have two sides. You have inside the structure, and then you're going to have outside of the structure. So anything that is enclosed within the cell membrane, or everything that is outside of the cell membrane. So I just want you guys to have a visual of those parts. All right, so let's talk about photosystems, because these guys are very important. So photosystems are reaction centers and light capturing complexes. So photosystems are pretty much made of two parts. You have the reaction center and you have the light capturing complexes. The reaction center is a complex of proteins associated with chlorophyll A and an electron acceptor. And then our light capturing complexes are pigments associated with proteins. Right, so that's pretty much what our photosystems are. We have two photosystems, so there's photosystem 2 and there's photosystem 1. Now, why do we have two photosystem 2 before photosystem 1? It's the order in which they were discovered. So this is just a backstory if you care. The scientists discovered photosystem 1 first, whoever that scientist was, and then later discovered that, hey, there's another photosystem, and they named that photosystem 2. So, hey, there are two photosystems. However, the one that was last discovered actually occurs, or it comes before the one that was discovered first. So if you're confused about that, and I'm just saying that because you guys need to know that photosystem 2 actually comes before, in the actual sequence of what it does, it actually acts or comes before photosystem 1. All right. Um, oh yeah, and then think of like antennas for reaction centers when you're thinking of light capturing complex. In the previous slide, so we have two photosystems, photosystem 1, photosystem 2. These are embedded in the thylakoid membrane. Photosystem 2 is a reaction center. Photosystem 2 is also named the six P680, and this is because it actually absorbs light at that specific wavelength. So it absorbs light at 680 nanometers, and that's the wavelength at which it absorbs light. Um, the photosystem 1, which comes after photosystem 2, is another reaction center. And remember, it absorbs light at 700 nanometers, and it is called P 700. And again, this was named in the order in which they were discovered. So this is some additional information about the photosystems. So they pretty much do the same thing. However, they're absorbing light at different wavelengths. All right, so now we're going to be looking at the steps. We're going to be looking into the photosystems, and then we're going to be looking at the steps pretty much leading to our final products of the light reaction. All right, so inside photosystem 2, so remember photosystem 2 comes before photosystem 1. So this is the photosystem for the sake of a diagram. So the photosystem is made up of the pigment molecules. So these are going to be our chlorophyll molecules. And then we're going to have a primary electron acceptor. And then remember that photosystem 2 absorbs light at the 680 nanometer wavelength. All right, so light comes in. So light hits the pigments. So remember, the pigments are able to absorb light. So photons hit the pigments, and this causes an electron to go from an excited state to a ground state. So it's going to go up, it becomes excited, and then it's going to go into a ground state. And this process is going to be repeated until it reaches the pair, that is the P680 pair of the chlorophyll A molecules. So what is happening is these molecules are, sorry, these pigments specifically, they're absorbing sunlight, and the electrons are becoming exciting. And as the electrons are now very excited because they have all this energy, they're hitting all the pigments, and they will keep doing that until they actually hit this P680 pigment right here that absorbs that excited electron. And then once it does, it becomes, it gets to a very high um, state of excitement, and then the electron acceptor will now accept that electron. So the electron is transferred to the primary electron acceptor, and this pretty much forms the P680 plus. So this is telling us that it's gaining an electron, so therefore it's becoming reduced. And it's pretty much an electron acceptor. So this molecule is accepting the electrons. All right, so once the molecule accepts the electrons, um, water comes in. So H2O comes in, and this water is going to be split. So this water is now split by this energy, and the water becomes, well, water is now oxidized. So we're going to have oxygen leaving, so it's losing electrons, and then we're going to have our nitrogen. So it says here H2O is split into two electrons, and that is used to reduce the P680. And remember what reduce means, because the P680 is the electron acceptor, it is reducing it by gaining more electrons. Um, so what we have now is, because hydrogen, the two hydrogens have now given up their 2 electrons. What we have left are protons. And these 2 protons will be released into the thylakoid space. And this is going to be building or proton gradient. One oxygen atom immediately bonds to another oxygen atom, and then that oxygen diffuses out of the cell as oxygen gas. I want to pause here a little bit to let you guys know that you have to think of a continuous flow of water coming in. So even though you're probably thinking, oh, when water is split, so when water is split into hydrogen and oxygen, this is just one oxygen. But remember, other molecules of water is going to be coming in. So the one oxygen molecule will wait for, and it's not a long wait, will wait for another water molecule to come in. The same splitting occurs, and then that oxygen will join another oxygen to form O2, and then that leaves as a all right, so we're gonna have like a linear electron flow. And what that means is that electron is pretty much gonna be flowing in a line, and it's gonna be flowing like in a specific order. So it's gonna be moving from photosystem 2 on its way to photosystem 1, and there are some other proteins or molecules that are in between that will be collecting that excited electron. And as the electron is moving from these electron acceptors on its way to photosystem 1, it is actually losing momentum. So it's actually losing some of that energy. And thus why photosystem 1 is very important at the very end, so that photosystem can also capture some energy to re-excite this electron. So the electron is excited. So as these excited electrons are being picked up by their primary electron acceptor, they're now leaving photosystem 2 and they're going to photosystem 1. And there are some molecules in between. You don't need to know these molecules, but just know that photosystem 2 and photosystem 1 are not direct neighbors. So there are other molecules in between that thylakoid membrane. So we have the PQ molecule. It doesn't matter if you don't have to know what PQ stands for, I promise you. And then we have cytochromes, and then we have PC. So the electron is going from photosystem 2 to PQ to cytochrome and then to PC, and then it's going to be going to our photosystem 1. All right, so this diagram should make things a little bit clearer. So let me go over this diagram with you. There's stuff at the end of the diagram that you probably don't quite understand yet, but we're going to go over the ATP, the synthesizing of ATP shortly. All right, so here we have our first photosystem. So this is our first photosystem that contains chlorophyll that is able to trap sunlight. As that electron or the electrons are being accepted by the electron acceptors, which are embedded in the photosystem, those excited electrons are being transferred from photosystem 2 to the PQ to the cytochrome, and then there is a fall right here. So here we have the PC, and then electron should go to photosystem 2, right? So this is the linear movement of the excited electrons. Now, the fall of the electrons from photosystem 2 to photosystem 1 provides energy to form ATP. And this is going to be a coupling reaction in which there is another molecule, or an enzyme should say. ATP synthase is responsible for synthesizing that ATP molecule. However, it couples that reaction with pretty much pumping out the protons or the positive hydrogens um, over on the other side of the thylakoid membrane. All right. So when we get to photosystem 1, we have the excited electron. So there's a fall, and then ATP will be synthesized, and this is going to be a coupling reaction, and I'm going to get back to that in a sec. So here we are at photosystem 1. pretty much the same thing will occur in photosystem one. We're going to have light coming in. So this is new light. And remember, light is going to be captured at a different wavelength for photosystem one. So photosystem one again captures sunlight in order to like pretty much re-energize these electrons. So light is going to be captured and it's going to be moving from pigment to pigment. Once it hits that P700, that P700 will be reduced, and then we're going to have our final or our primary electron acceptor accepting that electron. And then once those electrons or that electron is accepted, then that electron now goes to our final electron acceptor. So this is now our final electron acceptor, not a part of a photosystem, and that is going to be NADP reductase. And NADP reductase, as the name suggests, this is an enzyme. So NADP reductase collects these electrons, and then that is pretty much it's these electrons are now going to be transported over to our Calvin cycle. So NADP now becomes NADPH. So electrons go down the second transport chain, and then we have NADP reductase, which is an enzyme. It catalyzes the transfer of electrons from FD, and FD is like an intermediate molecule that collects the electrons, and then it becomes then those hydrogens, those positive hydrogens, become a part of NADP to become NADPH. And I want you guys to note the name of the enzyme. So NADP plus reductase, that is telling you that this enzyme is responsible for reducing NADP. And if we're reducing something, remember redox reaction, if you're reducing, it means that you're gaining electrons. And so at the very end, we're going to have NADP plus gaining a hydrogen. And even though several electrons are coming through here, we're going to have just each molecule will just gain one electron to become NADPH. And then here we have our NADPH. All right, let's attempt to put everything together. So here we have the details of pretty much what happens in the light reaction. So here we have our chloroplast stroma. And remember, the stroma is a fluid-filled space. And then within the stroma, we're going to have structures like the thylakoid embedded. So the thylakoid itself is made up of a membrane, so it's a double membrane structure. So here we have the thylakoid membrane. And very similar to the cell membrane, there are different protein molecules that are embedded in that in the membrane. And then on this side right here would be inside the thylakoid. So think of if this membrane completely surrounded the thylakoid, then this would be on the inside, and then where the stroma is on the outside of the thylakoid. So now let's look at our photosystems. So photosystems are responsible for capturing light energies, capturing photons at various wavelengths. So we have two photosystems. Photosystem 2 comes before photosystem 1. Photosystem 2 is responsible for capturing light energy at the 680 nanometers wavelength. And photosystem 1 is responsible for capturing sunlight at 600 nanometers wavelengths, or capturing light in general. All right, so here we have photosystem 2. Photosystem 2 will absorb light. The chlorophyll, specifically the pigments in the photosystem 2, would absorb light at the specific wavelength. That light or the photon will be used to excite an electron, and these electrons are existing electrons from water. So these electrons become excited. They will keep moving from from pigment to pigment until they get to or P680, and then they're going to go to their final energy, their final electron acceptor within that photosystem. Once it goes to the final electron acceptor, that electron is now going to be doing its linear movement, in which it's going to be moving from photosystem 2 to the PQ to the cytochrome. And then once it gets to the cytochrome, there's going to be a fall in energy. That fall in energy is going to cause a coupling reaction in which the the protons, and I probably should have explained this part first. So where are these protons coming from? So let's pause right here. So here we have our electron, and this is the fall of energy. Why do we have protons inside the thylakoid lumen? We have protons because remember, every time water comes in, the hydrogen is pretty much relieved of its electrons, leaving behind its protons. And those protons are pretty much being captured inside the thylakoid lumen. And as protons are being captured inside the thylakoid lumen, it's building a concentration gradient in which there's a difference in proton concentration on the inside of the thylakoid versus on the outside of the thylakoid. And as this difference in concentration is occurring, what will happen is that once we have that concentration gradient, diffusion of these protons will occur eventually. All right, so I hope I didn't confuse you there. So let's go back to where we are at this PC. So here we have our electron, right? And this electron falls. What will happen at this point is a coupling reaction in which these protons are now going to be pumped by ATP synthase. ATP synthase is going to do two things. ATP synthase is going to be acting as a proton pump that's going to be pumping out these protons. And as it is pumping out the protons, it's going to gain energy. So it's almost like this pumping of the protons is pretty much causing these wheels to turn, and it's gaining energy, enough energy, so that it can synthesize ATP by adding. So it's not creating a molecule from scratch. So what it's simply doing, it's using existing ADP, and it's just phosphorylating. So it's adding a phosphate to those ADP, and that's how we get this ATP molecule. So ATP synthase synthesizes ATP from ADP, but ATP synthase as an enzyme also is a proton pump by allowing these protons that are gathered in the thylakoid membrane to be released into the stroma. All right, so while that is occurring, we have our electrons here at our PC that needs to go to or photosystem 1. Now, so the energy level has fallen for all these electrons that are coming. Once they get to PC, the energy level has fallen. So what's going to happen is that photosystem 1 is going to absorb energy again. And this energy is going to be used to excite electrons. All the electrons are going to become excited once that energy is once that new energy comes in, and then it's gonna get to an excited state, excited enough so that the primary electron acceptor within this photosystem collects the electrons, and then that electron is gonna go to another molecule, ferredoxin. Don't worry if you cannot remember the name for this, you don't have to. The ferredoxin collects the electrons, and then the ferredoxin passes that electron to another enzyme, which is or ADP plus reductase. And ADP plus reductase is responsible for adding hydrogens to NADP. So again, I don't know if I said that right. Hold on. So NADP reductase is responsible for adding hydrogens to NADP. So that's what it does. And I'm sorry if I said something differently just now. So that's pretty much how at the very end of this reaction, we're going to end up with these products. And let me skip to the next slide. Right, so key things to note. So in our light reactions, um, these are inputs and outputs. So input would be water, so water is coming in, and then there's existing ADP within that thylakoid space, and then there's also existing NADP within that space as well. So these are the things, these are the inputs. So pretty much you can say that these are the reactants pretty much. At the end of the light reaction, these are the outputs. So these are the products. So oxygen is going to be leaving as a byproduct. This ADP is going to be synthesized into ATP by ATP synthase. And then we're going to have all the electrons that were collected will now be used to form molecules of NADPH, and this process will be catalyzed by NADP reductase. All right, let's do a quick summary before we hop over into the Calvin cycle. All right, so the light reaction converts solar energy into chemical energy. Chemical energy are in two forms, so there are two chemical products that are very important at the end of the light reaction, and that's NADPH and ATP, and we should know how those things are made. Water is split, and the water is used to actually provide our source of electrons and protons, so those hydrogens from water are used to provide the electrons that are going to be doing that linear movement from photosystem to photosystem. And then the protons are pretty much going to form that proton gradient, and protons become very vital in the process of that pumping of the ATP synthase. O2 is going to be released as a byproduct, or you can say it's a waste product for the reaction. Light absorbed by the chlorophyll drives the transfer of electrons and hydrogen ions from water to the electron acceptor called ADP plus. And ADP, I don't know if I said it in like the lectures, but ADP plus is considered to be the final electron acceptor for the light reaction. And that's also a reduction process. So once it is reduced, it becomes NADPH. ATP is generated by phosphorylation or phosphorylating ADP. So it's pretty much just adding another phosphate. So remember ADP, adenosine diphosphate, it only has two phosphates, and it's going to be adding the third one. All right, here we are, the Calvin cycle. Now, lucky for you, you don't have to memorize most of the details for the Calvin cycle, but for the light independent reaction, you do need to know a lot more details. All right, so the Calvin cycle is the light independent reaction, so it does not require light. So the Calvin cycle can pretty much occur at pretty much any time, during the daytime, during the nighttime, it doesn't matter. If light is there, it doesn't use it. So the products, as I'm explaining this, I want you to bear in mind the products from the light reaction, they will go into the Calvin cycle. And the Calvin cycle is the synthesis part of photosynthesis. So this is where we're expecting our high energy sugar to be made, and that's our glucose. So the Calvin cycle is a cyclic electron flow. And the reason why it is cyclic is a cycle, which means that the reactants that are coming in, they are pretty much like they're transporters. So they're dropping stuff off and they're returning to the light reaction. So our ATP is coming into the Calvin cycle as ATP and is returning as ADP. Our NADPH is coming into the Calvin cycle and is returning as NADP plus. So that's what makes it a cyclic process. And then we're expecting sugar to be produced at the end. And then this is where carbon dioxide also comes in. And remember, carbon dioxide is a reactant of photosynthesis. So it uses NADPH to reduce oxygen to sugar or G3P, and it forms net synthesis of one G3P molecules, and the cycle must take place 3 times. So in order for us to generate one molecule of sugar, then this cycle must take place 3 times. All right, I'm going to go over the Calvin cycle in detail. And then when we get to class, I'll discuss with you like what you don't need to know. If you have the capacity to like go through the details and understand it, I suggest that you do, even though it's above the scope of AP Bio. But the more you know about the processes, the better it is to understand. It's like knowing the little things in order to just understand the big picture. All right, so there are three phases of the Calvin cycle. Um, phase one is carbon fixation. Phase two is reduction, and phase three is a generation of RUBP. All right, so let's get into phase one. So this is carbon fixation. So in carbon fixation, as the name suggests, carbon dioxide is going to be fixed to something. So carbon dioxide is coming in. So carbon dioxide as a reactant of photosynthesis is going to be incorporated in the Calvin cycle. One at a time. Each carbon dioxide molecule attaches to another molecule that is called the RUBP molecule. And this RUBP molecule, or this process of attaching carbon dioxide to RUBP is catalyzed by an enzyme that is called Rubisco. All right, so once we have the formation of the carbon dioxide molecule added to RUBP. And RUBP is a 5-carbon compound, and each carbon dioxide molecule has 1 carbon. So together they're going to form a 6-carbon compound. And that 6-carbon compound is unstable, so it's going to quickly break into two 3-carbon compounds. And each of the 3-carbon compound is phosphoglycerate. And as phospho suggests that it has phosphates, and the glycerate is suggested. All right, then we have our second phase, which is reduction. And remember what reduction is. If something is being reduced and we're expecting it to gain electrons. All right, so each molecule of this, each of the 3-carbon molecule, which are the phosphoglycerate, is going to be phosphorylated by ATP. And it uses 6 ATP molecules in total. So each molecule will be phosphorylated, and then each molecule now becomes, because it is being reduced, and remember what phosphorylated means is gaining a phosphate. So the ATP molecules that were synthesized in the light reaction are now coming over to the Calvin cycle, and what are they doing? Each of these 3-carbon compounds, they're pretty much adding a phosphate to the 3-carbon compound. And by doing so, they're reducing the compound, because now the compound is gaining electrons, and then also it's becoming phosphorylated because they're pretty much giving them a phosphate. So by gaining a new molecule, what's happening is that this compound has now changed. So this compound is still a 3-carbon compound because it did not gain or lose carbon, but it's a new compound. It's now called the bisphoglycerate. All right, so and then here we're gonna have, or 6 molecules of NADPH is gonna come in. So 6 molecules of NADPH is actually required, and the 6 molecules of NADPH will now donate electrons. And remember, electrons are in the form of hydrogen to one um, 3. So it's to one of the 3 bisphoglycerate compounds. And by doing so, it is reducing it to another molecule. So this is it's being converted to another molecule that is simply nicknamed the G3P molecule. So we now have 6 molecules of this G3P molecule are now formed, but only one is counted as a net gain, because we're gonna have to pretty much send one back to form that ribulose biphosphate that we started out with. Um, so the other 5 molecules are going to be used, yeah, I just said it, to regenerate ribulose biphosphate. So that's RUBP. And then the last phase is the regeneration of ribulose biphosphate. So RUBP stands for ribulose biphosphate. So we have, remember, we produce 6 of these G3P molecules, and we are going to be pretty much using 5 of the 6 to regenerate our ribulose biphosphate. And remember, if you recall, when we started out with the carbon fixation, ribulose biphosphate is a 5 carbon compound. So we need this 5 carbon molecule to pretty much regenerate this ribulose biphosphate. And then we have one G3P molecule um, that's just going to be used, or it's going to be waiting to form our sugar. It uses 3 ATP molecule for the regeneration. And where are these ATP molecules coming from? They're coming from the light reaction. The cycle is now ready to take in carbon dioxide again. So once this ribulose biphosphate has been regenerated, then this ribulose biphosphate can be combined with another. All right, so let's try to put everything together using a more like of a visual diagram. So I want you guys to bear in mind um, all the products from the light reaction. So we have ATP coming in. We're gonna have NADPH coming in, and then also we're gonna have carbon dioxide, which is coming in from the atmosphere. And remember, carbon dioxide is one of the reactants for photosynthesis. So here we're gonna have our ribulose biphosphate, so that is our RUBP. And let me try to write some stuff on here. Probably not, no. All right, so RUBP is a 5 carbon compound. RUPP will be fixed to carbon dioxide. So carbon dioxide is now going to be bonded with this RUBP to form a 6 carbon compound. This 6 carbon compound is a very unstable molecule, and therefore it quickly, as the minute it is formed, it quickly divides into 2 molecules, each having 3 carbons. So one molecule of RUBP combined with carbon dioxide forms a 6 carbon compound that divides very quickly into 2 3 carbon compounds. Each 3 carbon compound is called phosphoglycerate. And here we have one over here and one over here. And that is pretty much the end of our carbon fixation phase. And then next we're going to go into the reduction phase, which is phase 2. These 2 molecules individually are going to be reduced. And how does this reduction process occurs? So we're going to have our ATP molecule coming in. And where is this ATP molecule coming from? It's coming from the light reaction. So ATP comes in, and ATP gives a phosphate to each of the molecules. So that ATP drops off the phosphate, and that ATP now becomes ADP. Um, because this molecule has now gained a phosphate, the molecule has been reduced, and therefore this is the reduction phase. The molecule having a phosphate now added is not the same as our molecule, that our 3 carbon compound molecule, and therefore it is a new molecule. So this is now a bisphoglycerate. So the bisphoglycerate is further reduced by now gaining the hydrogen from the NADPH. Remember, NADPH is coming from the light reaction. So this molecule now gains a phosphate. This is still a 3 carbon compound. So even though it is being reduced and we have phosphate being added and now we have hydrogen being added, it still only has 3 carbons because it has not gained any carbon. So it's a 3 carbon compound. However, now it's a different compound. So here we have the G3P is what it becomes in the end. So remember that we had um, 2 of the 3 carbon compound. As you're tracking this cycle, we're only discussing one, but remember the same thing is happening to each of them as it's going along in the cycle. So here we are, we have our G3P molecule. It takes 5 G3P molecule to pretty much regenerate ribulose biphosphate. So in the regeneration process, remember that ribulose biphosphate is a 5 carbon compound. So we need that carbon compound to be regenerated before we even think about making sugar. So once we have six coming around, six carbon compounds, five of those will go back to form the ribulose biphosphate, and then we'll wait until more molecules are produced in order for us to be able to form or glucose, which is a six carbon compound. So pretty much this is the details of the Calvin cycle. AP Bio does not necessarily want you guys to know all that detail, so you don't have to know specifically. Um, and I don't want you guys to quote me on this. I really want to go back and read that document. I do believe that ribisco is important, and ribisco is the enzyme that is responsible for fixing ribulose biphosphate to carbon. So I think they require you guys to know that, and then you also have to know what happens to each of the products from the light reaction. So where do they go? What do they do? And how are they sent back in the process? You do not have to know the details of what molecules, any name of these molecules. If these names are causing you headache, or if you're tripping over these words, it's good because you don't have to know the names of it. But Ribisco is the enzyme that I think you guys need to know this enzyme, and also you need to know what happens to all of these products from the light reaction. Similar to what we did for the light reaction, we're going to do the same thing for the Calvin cycle. So here are the inputs and the outputs. So we need three carbon dioxide molecules in order to form, to regenerate or ribisco, and also to form our sugar. So carbon dioxide is needed. In total, we're going to need a 9 ATP molecule and 6 NADPH molecule in order to regenerate a carbon, sorry, a glucose molecule and ribisco and ribulose biphosphate, sorry. And then the outputs, what are we getting in the end? We're going to have one G3P molecule, and that's after the ribulose biphosphate has been regenerated, and then we're going to have a 9 NADP going back to the light reaction, and we're going to have 6 NADPH plus going back to the light reaction as well. And here is a summary of the Calvin cycle. Um, Now, there are some problems with plants that photosynthesize this way. And plants that photosynthesize this way are called C3 plants. And there are some issues with the C3 plants, and therefore plants have pretty much evolved to not photosynthesize in this way. So here's a problem. There's a problem called photorespiration. Because photosynthesis and cellular respiration are almost like reverse reactions of each other, um, the stomata is responsible for gaseous exchange, and it's that said stomata that will allow oxygen to leave as a waste product of photosynthesis. However, that said oxygen is also vital for cellular respiration. So, oftentimes during hot days, the stomata closes, and this is an adaptation that plants have. The stomata will close in order to prevent the loss of water, and by closing, it's also preventing gaseous exchange. So it causes less carbon dioxide to come in. However, there will be a buildup of oxygen because oxygen is being produced inside of the plant during photosynthesis. Carbon dioxide is external to the plant, and that needs to come in from the atmosphere. Um, Ribisco binds to oxygen and uses ATP. So the ribisco is our enzyme that we talked about, and ribisco will cause ribulose biphosphate to pretty much bind to oxygen. So there is really no preference for carbon dioxide over oxygen in the Calvin cycle. And therefore the oxygen will bind to the ribulose biphosphate, very similar to how carbon dioxide does. So in the event that the plants have their stomatas closed and there's no carbon dioxide coming in and there's an excess of oxygen being produced, what will happen is that excess oxygen will pretty much compete for this ribisco or the ribulose biphosphate. And once this happens, it pretty much halts the photosynthesis process. So no sugar is going to be produced. So in the Calvin cycle, if oxygen is fixed to ribulose biphosphate as opposed to carbon dioxide, it means that the plant does not produce sugar. So no photosynthesis will be produced in the process. And that's a problem for plants if they're not actually producing their desired products at the end of photosynthesis, which is glucose. That's why they're investing all this energy into this process because they want food at the end. So plants have evolved to bypass this product, or there are mechanisms in place to prevent that from occurring. So here are some of the adaptations that plants have. Plants that live in arid conditions, which are hot and dry climates, they have evolved to form like alternative mechanisms or ways of fixing their carbon dioxide. Um, so these plants are categorized as C4 plants and CAM plants. C4 plants are designed in a way that they spatially separate the steps in photosynthesis um, because their stomata are partially closed to conserve water. So within the cells, and I have a diagram of what the cells actually look like. So the mesophyll cells, they fix the carbon dioxide into a C4 molecule, and that's why they're called C4 plants. So the C4 molecule is oxaloacetate, so they will fix it into the C4 plants, and then they will fix it in the C4 molecule, and therefore, and that C4 molecule will be fixed when it's time to or carbon dioxide. Um, and then they transfer this in a bundle sheet. So they're pretty much separating the process um, into like actual different cells in order to pretty much bypass this product. And examples of C4 plants are like corn, grass, and sugarcane. And then we have plants that are called cam plants. And these cam plants, um, they are more temporal. Um, for like a better description. So they open their stomatas only at nights. So cam plants live in like really extreme conditions in which they don't partially open their stomata. They don't open their stomata sometimes, but their stomata is always closed during the day. And then they only open their stomata during the daytime, which is a problem because remember, photosynthesis actually, the light reaction actually needs to occur during the daytime in order for us to get those products for a Calvin cycle. So CO2 is captured during the nighttime, but CO2 is incorporated into an organic acid and is stored in the vacuole. So it's temporarily stored in the vacuole during the daytime. And as the light reaction is occurring, at the end of the light reaction, that CO2 is going to be released from the organic acid and it's going to be incorporated into the Calvin cycle. And these plants are usually acidic to taste. So, like for example, pineapples and cacti and other like succulent plants and like jade. Those are examples of C, sorry, cam